Hello and welcome everyone. So this lecture is mainly for PDCT, Pediatrics and NEET PG students for Pediatrics topic. But in the coming month, PDCT Pediatrics exam is there. So we are trying to cover few topics which are important in exam point of view. So today's topic is disease of mitochondrial inheritance. So mitochondrial disease, and this is a small but important topic, and many times MCQs are put up from this topic, so we will try to read this one. So mitochondrial diseases, first of all, something about mitochondrial disease, a brief introduction we can say. So what are the mitochondrial diseases? These are the group of genetic diseases. These are the group of genetic disease. Important point is genetic disease, and what is happening caused by defects in OXPHOS. OXPHOS means oxidative phosphorylation. Oxidative phosphorylation. So the defect of oxidative phosphorylation. So what is the defect? Defect in oxidative phosphorylation, and due to, another line is due to mutation. Due to mutation in, now this is important, mutation in what? Mutation in the mitochondrial DNA or the nuclear genes. The nuclear genes encoding what? Encoding mitochondrial proteins. Okay, so now these are the group of genetic diseases. If we go through this again, then genetic diseases caused by defects in oxidative phosphorylation and due to mutations in mitochondrial DNA or nuclear genes encoding mitochondrial proteins. So this is what mitochondrial disease is all about. So now moving on to next topic, that is basics of mitochondrial disease. And in basics of mitochondrial disease, we will look at the important clinical features. So what are the important clinical features for any mitochondrial disease? First of all, few important points, that is maternal inheritance. What is the type of inheritance? It is maternal inheritance, that is uh, inherited from mother, only mother. Okay, this is important in MCQs. If they give history, then only mother can transmit this disease. So this is known as maternal inheritance, and mitochondrial disease is a classical example of mitochondrial inheritance. Other point, key feature, we can say this is basically not the clinical features but important features for this mitochondrial inheritance. So another one is mitochondrial DNA. What is the shape of mitochondrial DNA, or characteristic feature of mitochondrial DNA are circular. So remember this, mitochondrial DNA are circular. And how many genes do they encode? They encode 37 genes. Encodes 37 genes. And the other important feature is, this is the very important one, that mitochondrial disease affects the tissues with high energy demand. So predilection for the tissues with high energy demand in case of mitochondrial diseases. So which are the tissues with high energy demands? Let's see. Tissues with high energy demands. Examples. First, very important one is brain, muscle, heart, retina. So these are the important tissues with high energy demand. So these are mostly affected in mitochondrial diseases. Okay. So many times questions are framed like this, that in which of the following disease high energy demand tissues are most likely to be affected. So mitochondrial diseases. Okay. Now moving to next is something about the genetics of mitochondrial disease. So what about genetics of mitochondrial disease? First, as we have already read in the previous slide, that maternal inheritance. What is the type of inheritance? Not autosomal recessive or dominant or X-linked. It is maternal inheritance, and all children of affected mother. So if the mother is affected, then all children are affected. Okay. And fathers, what about fathers? If the father is affected, father do not transmit. This is very important. Do not transmit this disease. So this is maternal inheritance. Next point is heteroplasmy. Now, what is heteroplasmy? Anyone? Heteroplasmy is that the cells contains both mixture of नॉर्मल एंड म्यूटेंट माइटोकॉन्ड्रिया. ओके, वन सेल इज देयर, एक सेल है उसके अंदर नॉर्मल माइटोकॉन्ड्रिया भी होगा और म्यूटेंट माइटोकॉन्ड्रिया भी होगा. सो दिस इज नोन एज हेट्रोप्लाज्मी. हेट्रो मींस मिक्सचर. सो नॉर्मल एंड म्यूटेंट mitochondria is a feature of mitochondrial diseases. Fine. So another important point for unique concept about this genetics of this mitochondrial disease is threshold effect. What is the threshold effect? Symptoms appear only when mutant load crosses a critical level. That is, threshold effect is all about critical level. Threshold effect means a particular like suppose for a viral disease we can say that for particular viral load, if that viral load is achieved, then that disease appears. So in this case also symptoms appears at some critical levels. Like for a particular disease, if 100 is the critical level, that is the mutation or mutant load should be 100. So if it is more than 100, then disease appears. If it is less than 100, the mutant load is less than 100, then disease doesn't appear in that particular case. So this is again a feature of mitochondrial diseases, and variable expression is also there. So this is about uh, if we can add then variable expressivity. In some person, the disease is expressed more aggressively, while in other it is expressed in a mild manner. So variable uh, expression is another feature of mitochondrial inheritance. So this was about genetics. Now, if we go to the pathophysiology, what is the pathophysio behind these mitochondrial inherited diseases? So pathophysiology, mitochondrial dysfunction. Now, first of all, what is happening? Mitochondrial diseases. So mitochondrial dysfunction. And mitochondrial dysfunction is leading to what energy source of the cell is mitochondria. So if mitochondrial dysfunction is there, then there is decreased production of ATP. So if decreased production of ATP, what is ATP? ATP is the source of energy. Energy is formed in the form of ATP only in our cells and bodies. So if the ATP gets decreased, then it affects what? It will affect the energy dependent tissues. Okay, it will affect the energy dependent tissues. So what is the mechanism for this mitochondrial dysfunction? Why ATP decreases? First is defective ETC. ETC means electron transport chain. And then what is happening because of this defective electron transport chain? Increased level of lactic acid, because lactic acid production takes place in anaerobic metabolism. If the ETC is not functioning properly, electron transport chain is not functioning properly, mitochondrial dysfunction is there. So increased lactic acid production because of anaerobic metabolism. And what this does overall, this increases the oxidative stress inside the cells or body overall. So it increases the oxidative stress. So these are three mechanisms which are occurring in the pathophysiology of mitochondrial disease. So what is the hallmark basically? The if we say what is the hallmark of mitochondrial diseases, so lactic acidosis can be considered as hallmark. If in any clinical scenario, if lactic acidosis is given, then we can suspect mitochondrial disease in that particular patient. So now this was about basic pathophysiology. Now if we take examples of some mitochondrial disease, then what are the examples? The first one is MELAS. Everyone might have heard about this MELAS syndrome. What MELAS stands for? MELAS stands for mitochondrial encephalomyopathy. Encephalo means brain, myopathy means muscles. So ME stands for mitochondrial encephalomyopathy. Now this L stands for LA stands for lactic acidosis, and this S stands for stroke-like episodes. Okay, so if you are given a case scenario that an 8-year male or female is presented with encephalitis like changes or muscular weakness and lactic acidosis is seen in blood workup, and MRI is suggestive of stroke-like lesions in the brain. So you can suspect MELAS syndrome in that case. And what the type of stroke which is for happening in this? So the important features, the clinical features of this MELAS is stroke-like episodes in young age. Okay, so this is very important. Young stroke mostly. Young child or young adult will present with stroke, and so young stroke less than 40 years of age. So MELAS is suspected. These patients presents with seizures because of encephalopathy-like features. So seizures may be there, headache and vomiting, headache and vomiting, lactic acidosis. Another one is lactic acidosis, and short stature can be seen. These are the main clinical features of MELAS, and the important point in this is that stroke-like episodes, but stroke does not follow any vascular territory. It doesn't follow vascular territory like in adults in normal older population. When stroke is happening, then it is following a particular vascular territory like middle cerebral artery infarct or posterior cerebral artery infarct or anterior cerebral artery infarct or posterior circulation stroke. But in case of MELAS, this territory is not followed. So if you find a young child with stroke, and that stroke is not following a particular territory in MRI imaging, then you can suspect MELAS in that case. Okay, so this was what disease? Now another example is MRF, that is MERRF. This is another important disease, and what is the full form? Myoclonic epilepsy with ragged red fibers. So this is important, myoclonic epilepsy with ragged red fibers. So what are the clinical features? Myoclonic seizures, as the name is suggesting. Myoclonic seizures are seen in these patients. Patient may present with ataxia, and patient may also have dementia. So these are the features of myoclonic epilepsy. Ataxia and dementia are the features of MORPH. What is the basic pathology seen in this disease? When you do muscle biopsy in the suspected cases, you are supposed to see what you are supposed to see, as the name suggests, red fibers. Okay, so red fibers are seen. So this is in short about MRF. Now another